Hola, muy buenas tardes a todos y
Good
bienvenidos.
afternoon and welcome to this Science Café. My name is Cesma and I have the chance today of introducing you to these excellent speakers. Thank you very much to those of you who are here today on site and also those of us who are following us online. Today we'll have the opportunity of discussing a very important topic, i.e. the data that are obtained to develop therapies and to develop clinical trials that will be the base for future therapies and how clinical trials approach the topic of inclusion. We know how important it is to have safe data and effective data. However, we also know that this data doesn't cover all the population groups that we have in society. Usually, this data is very specific, and very often we have excluded some immunosuppression population groups or children. And when we exclude this data, it means that the therapies developed are not so inclusive in terms of including all the population groups. We want all the population groups to be included in these clinical trials. And for this, we have three excellent speakers. Firstly, Dr. Beatriz Mothe, who is the coordinator of the RBDCOV trial on immunosuppressive conditions that is associated with the Germans Trias y Pujol Hospital. We also have with us Dr. Pere Soler, who is the principal investigator of the clinical trial on the HH3 clinical trial on pediatric infectious diseases. And we also have Joan Tallada, who is a member of the RBDCOV Community Advisory Committee and is a representative of the European Treatment Group, and who will bring the community and patient perspective to these processes. Thank you very much, Laura, and I'd like to thank the European team for inviting me here today. I have been invited as an investigator in a clinical trial that is implemented specifically on individuals with immunosuppressive conditions. And as Laura mentioned, we know that there is a lot of inequality in clinical trials because many clinical trials are developed for healthy populations, and then the therapies are provided to populations who are unhealthy populations. This is something that we already saw with COVID, and it has many different reasons, demographic reasons. Maybe we don't ever represent all the ethnia and race differences. We may also see some inequity due to gender or age. The same happens with children, the elderly, women. There are also some baseline conditions, immunosuppressive conditions, that can be also a gap in many trials, and also geographical and social cultural aspects that are often not taken into account. And this leads to some inequity in the clinical trials. And what are the consequences? Well, if we don't do a clinical trial that includes a diverse population, the results cannot be interpreted in a general way for all of the population. So in this way, we have some inequity in the interpretation of the data. And when you try and implement the treatments in other populations, these treatments are not so safe and they're not so effective and don't provide the same outcomes as we see in the clinical trials, because we have just focused on a specific population. And this is an inequity that is seen also in the healthcare system. And I do believe that We need to solve this because this will cause also some mistrust in those who receive a treatment when they see that the treatment hasn't been tested in their own type of population. In the case of RBDCOV, we thought it was very important to look at the immunosuppressed population because we saw that with SARS-CoV-2, we realized that the tested vaccines were tested in populations that had no immunosuppressive conditions. However, the use was approved for those populations. We know that there is a large immunosuppressed population. There may be some baseline conditions, and there are also some treatments that can cause immunosuppression. And if you want to design a clinical trial that includes this immunosuppressed population, it's important to consider all these aspects. Here we have the effect of immunosuppression in the context of COVID-19. There was a higher risk of acquiring the disease, of having a more serious disease, and of having the disease for longer. And also there was a higher risk of developing mutations. So the immunosuppressive population was a key population to test the vaccines on. And the goal of our project, of this RBDCOV project, is to include also this immunosuppressed population. I'd like to give another example, i.e. the children population and the adolescent population that are now included in studies, but very often we see we have to wait for the adult studies. In the case of SARS-CoV-2, things were different because the measures applied to adults weren't necessary for children. Unlike what happens with the immunosuppressive population, we have many challenges in pediatrics research where we can have also immunosuppressed patients and baseline conditions. And the most common problem and the most difficult to solve problem is the capacity of including patients in the clinical trials. Most of us are parents here, and we are less reluctant to participate ourselves in a clinical trial than having our children participating in a clinical trial. When there is just one single treatment that's an option, then it's easier to have them included in a clinical trial, and it's more difficult to have healthy children also included and to have this approved by the CEIM, by the ethical committee. Of course, when you have to schedule the appointments for children, you also have to bear in mind the schedule of the parents and of the children. And there's always this fear of the parents to include their children in clinical trials. And there is also this fear of invasive treatments. It could just be a puncture. So any of these processes with children will always last longer. But inclusion is always more difficult. This is one of the main challenges because of the short and long-term toxicity, because of the invasive tests. And very often we see that treatments don't have the right dose for children. And then the response to treatment is usually better defined for the adult population than for the pediatric population. And this can lead to wrong conclusions. There is also a challenge with regard to the structure of the clinical trials for children, because very often the pediatric context is not well known. Very often what happens in a clinical trial for children is that they take the clinical trial for adults and then just apply it directly to children, just changing the age. We have fewer centers. We also have fewer networks. Very often we need new networks, and many of these centers have less experience and fewer resources. And very often we have children mixed with adults. And that's why in the end, with children, we have lesser quality studies, and that lag behind the adult studies. How should we approach this? We need to promote pediatric research centers at a local level. Here we have some examples of what we have done at our own center. And the main thing is to create a network, because in the case of immunosuppressed patients, the number of patients that we can include in the clinical trials is lower than with adults. And we have, for example, the C4C network at a European level, with which we participate actively. And in this way, we try and solve the situation of inequity that was mentioned before. Now I will give the floor to Joan. Thank you very much. In this project, as in many other projects, we have a community advisory committee, and I'm sure that many of you here already know about this type of committee. The idea is to have people representing the patients, interacting with physicians and researchers, so that the perspective of patients is taken into account in the design and the implementation and publishing of the research. I would like to tell you a story, because this is not something new. This is not something that has just happened overnight in the last 5 or 6 years. It does have a long story, and I would like to explain my own personal story, because we have to remember that this was a collective struggle, and some of them have already passed, some are here, some are in this room with us, some just left. And I like to explain my own personal story. I like to explain how I experienced this and how we reach at this point. Here we have 2 images. Firstly, we see an article of a paper article in the 90s. We see a photo, and in this photo, I am speaking about a group that I belong to, and it was one of the first times where we started as lay people to understand medicine. We would receive books and documents published by other members of the community on HIV. We wanted to understand HIV. We wanted to understand what was happening, and that was before the antiretrovirals appeared, where there were no viable options, and many people would get sick and die. In 1993, we had the antiretrovirals. In this other photo, I am with a colleague of Portugal, and I remember that this was one of the first meetings I went to in Frankfurt, where we started to discuss antiretrovirals with the northern European companies, because in the southern European countries, Spain, France, and Italy, we would insist on pharmacokinetic studies with methadone, because if not, we couldn't have included in the clinical trials those who were taking opioids. Because once the drug was approved, We achieved this, and the regulating companies understood that this was necessary. So now we're going to move on to the next slide. And in Sitges, this is a town near Barcelona, where different meetings were organized. Once antiretrovirals were available, we saw that many patients were dying due to hepatitis C. We saw this paradox after struggling for many years to have access to these research studies on antiretrovirals. Then patients would die of liver failure, and very often there were no organ transplants for HIV patients. That's why we wanted to accelerate the access to the new antiretrovirals for hepatitis C. I remember very well these meetings because I remember talking to the companies, to the pharma companies that weren't used to talking to patients, and they were amazed at the fact that we were asking for studies in a vulnerable population.
Because they were afraid that this would give many negative data against that drug. But we argued that there was no other option for these people to access these antiretrovirals, and then that we would oppose to marketing of the drug unless these studies were done, and that nobody like us would fight for these drugs to be approved. And the regulator was very doubtful about this. It took us 3 annual meetings to get to bring everyone around the table so that they would change their mind, so that these studies of hepatitis C antiretrovirals was done with people who were HIV positive before marketing of the drug, and that was approved. I have some pieces of some research that I participated myself as a representative of the EATG. This is just another example. Giorgio Apostolos and many other colleagues who are probably connected online, they are probably part of this exercises where we discussed with researchers who were studying our community, and we were discussing on an equal footing. And if we did not understand something, we would ask for it, because medicine is something that you study, and if you study, you can understand it. So that's why we thought we really needed to be involved in this. That's why for us, this has been a very long road, but there's still a lot to be done. There are many areas in medicine where, as has been said, special or vulnerable populations are not taken into account. I wouldn't even say minority population, because if you eliminate all those who are minority populations, I'm sure that nobody's left, because the group that would be left would be as small as any other minority. So the Belong Project fights for the inclusion of HIV-positive people in non-HIV clinical trials. We want to be included in cancer trials and everything that has to do with aging, especially neurocognitive disorders, because the cohorts of people who live with HIV, thanks to antiretrovirals, are aging as well. And we're seeing that some of the traits are accelerated because the brain is a reservoir for HIV. So even if the antiretroviral treatment does work, there's also some activity. And that happens as well for COVID and for other diseases. And there are some challenges we face here. One of them is lobbying the pharma companies, and I will say that very bluntly. Unless there is a financial incentive, they won't do it. So in the project we are involved in, this RBDCOV, who is an EU-funded project, So private capital did not want to be involved in this, and we needed to have public support. Everyone got really involved and everything was fine, but it's essential to take that into account, and we really need to defend the role of the public sector, because not everything can be in the hands of the market, and we will not be able to introduce those traditionally excluded groups unless there's a strong support by the public sector. And that requires regulation to be clear about the kind of data that need to be available before any drug is approved for marketing. And then let me say that this lobbying is not only for society and other stakeholders, but also with other patient groups, because for better or worse, the HIV cohort comes from a very specific background, and it's not always easy. In our project, it wasn't easy to involve representatives of people who were immunosuppressed because they had transplants, for instance, or parents of teenagers, because they don't have that tradition. So it's a little bit harder for them to understand the value of this, and that requires time and quality time. It's not something you can do after hours, after work, and after having done other things, because this is a full-time job, which is complex and requires a big commitment, but it offers great social benefits. It improves science, and it gives more purpose and more social and public value to the investment that is made, which in this case is clearly a public investment. And I'm seeing here my colleagues, Apóstoles and others who submitted this project with Brian and our close friend Luis from Portugal in a conference in Glasgow, a scientific conference in Glasgow. They presented the project there. And we will keep on fighting so that the way science is done changes, so that no one is left behind. Thank you. I think, as we have said, we have discussed what is important for clinical trials, because in the end, the purpose is to generate data on efficiency across the population, because very often those therapies will be targeted to these groups of people. So we need to understand better the feasibility of and the impact of those drugs in underrepresented populations. We need to put people at the center and not researchers at the center. We also want to mainstream results. We want to reduce the inequity in the healthcare system. If we are if we know the people we will treat better, we will treat them better. And this also contributes to generate more trust across the board by the general population. So this is a project where lots of people participate, not only promoters, those who will design the protocol with the help of researchers, but it's important to include the community or communities, as Joan said, when there are communities of patients or people who live with those conditions and that are already structured as a group, things are easier. So we need to work on that. And it's also important to work together so that the regulator can help us in this, because regulators don't change the paradigm. They want to test things first in X population, and then once it is approved and so on. But if the regulator would ask, who is your target population? If your target population is that kind of groups, then why not force to have information from those groups as well? At least in parallel, COVID helped us understand how to do adaptive designs, easier designs that did not have that sequential structure. So phase 1, phase 2, phase 3, and then tests in special populations as we are used to. This could have been much more, well, it showed that we could be much more creative in designing those inclusive trials. So we should all be working together to create these more inclusive clinical trials and generate data in these groups of population. And I think that so far, this is our part. And now we should give voice to the rest of our colleagues. Thank you so much to the three of you. I thank you. And I hope that the audience, both here and those connected online will take advantage of this opportunity to have people here. We have remote connection by Dr. Petra Wülfroth. Sorry if I didn't pronounce your word correctly. She has a great experience in the technological project, and she's the chairperson of PCO Technopharma from Germany, and she's experienced in the COVID project, and she will bring her vision. Thank you for being here. You have the floor, madam.
Okay, thank you very much. And you actually pronounced it principally correct. Could I have my first slide, please? Okay, thank you very much. I would like to share our experience, which is it's really connected with the pandemic, but it's also quite important in other situations. And when I was listening now to your talks, I thought now we are coming from a different direction. We are targeting the host-directed, we are looking at the host-directed therapies. Next slide, please. When we have a pandemic situation, we have different options. We could use antivirals, we could use antibodies, we could use vaccines, and we could target the host. And I think this is an area which is kind of neglected. When you're thinking about the different approaches, of course, it is always most important to have direct attack against the virus. But on the other side, and this is something what COVID taught us quite sadly, partly, that there are situations when there are no drugs available, there is no vaccine available, and there we really have to concentrate on host therapy. And I would shortly talk about FXO6. It's a peptide which we are developing in phase 2 in COVID. Next slide, please. So looking again at the host-directed therapies, in situations where you do not have direct therapy, you could support the patient with a host-directed therapy until the patient's own immune system is upregulated. It can also support antiviral therapies. This happened only recently with Ebola, that there are drugs against Zaire Ebola, but not against Sudan Ebola. So having something as add-on. The same is true for vaccines. There are vaccines available for certain infectious diseases, but sometimes with limited efficacy. So there is not something else needed on top. And it's, of course, also possible to support vaccinated patients with a pure immune response. And it could be in situations where there is really an absolutely unknown threat first kind. Next slide, please. So what I want to show to you is a COVID project. Very shortly, this was started during COVID. It was a quite large consortium, and it took us quite some time to include all the different sites and countries, and it came out that we were not able to have enough patients because, fortunately, at that time COVID was over. Then we were thinking about what could we do, because we have to be prepared for the future, and we switched, and the EU fortunately agreed, from a study concentrating on COVID, patients having COVID and AIDS, acute respiratory distress syndrome, to a study with AIDS of different sources. So including COVID-19 patients. So we were able to switch the study from a pure COVID to a mixed study. And this study is ongoing, and it's in the focus is a clinical trial phase 2 with a peptide called FXO6. And around there are different smaller studies, in vitro studies, and also a very in-depth omics approach, which we are very looking on the different proteomics, lipidomics, and metabolomics. And we hope that we can share results in the near future. Thank you very much.
And I think now that we've had the possibility of listening to these wonderful talks, on populations that are usually left outside of these trials, and the need to have all kinds of participants.
Para que se puedan hacer ensayos clínicos representativos.
I have a few questions, but I hope that from the audience, you also have some questions that you would like to ask and get clarification for. So let me open the floor for questions first. We have a question from Carla Aguilera. Thanks for your interesting presentations. It talks about the current needs for clinical trials, and thinking about protocol designs, I think it's important to involve patients' organizations who can provide a more human and less scientific approach. Would you say that we could improve their role in study design? She's a manager of the Neuroblastoma Research Association in Europe, and she was also involved in the Vall d'Hebron Pediatric Hematology Study Task Force. May I go? Yes, of course. As Joan said, it is increasingly frequent to have groups of people who are not part of the healthcare system, so they can provide their opinion on how we do research. And this should be much more mainstream. But we have already integrated this, and we actually have, for instance, teenager groups who participate in this. But the difficulty is to go to the very onset, how to design the study, because as Joan said, mostly these clinical trials have a private promoter, which is the pharmaceutical industry. And obviously, the participation of people who are not linked to that, patients or patients' organizations, usually delays the whole process, the whole trial process, because first of all, they require training. They are very demanding on things which are not included, and they probably add demands that are closely related to their subgroup of the population. And I would like to know what Joan thinks about this, but I guess that probably this is a step we need to make if we want to involve citizens in research, because very often we play to the gallery, but I think that unless we go to design, where we have less possibilities to ask for the participation of citizens, we will not go much further. What do you think, Joan? I think the key is to try and help make understand the pharma industry that they will end up benefiting from this, even if it takes longer, or even if it costs more money, because that's what they really care about. They'll have to sit down with people, to take it slowly, to give explanations, to listen to others, and maybe their marketing plans will not be 3 years, or maybe 4 or 5 years down the road, but they will be better because this drug will be much better accepted by the market. We've often been asked to review protocols that were already approved because it's obvious that this is not going to work because it wasn't well designed, or sometimes we are asked to help in the recruitment phase because it was not well designed, and then it means it takes much longer. So I know this is a cumbersome process, and there's more demands on the table. But I wish I could put an end to capitalism. But I mean, that's not my role. I'm there to get the drug to the patient within the system that we have, which is what we have. So I need to understand that you have certain needs as a private business, but you also need to understand that those needs need to match the social benefit too. And in the end, this will improve the final result. In the case of hepatitis C antivirals, the first patients in this country were infected people, the very first ones. We were the ones who were asking for this access. The jointly infected were asking for access as well, and we had to close an alliance with them. But it's not the same to have a chronic disease that have a treatment than a disease that can be cured. Because in the case of hepatitis C, we had those temporary alliances with the groups, but as the critical mass of people with hepatitis C disappears, because fortunately, those drugs offer a functional cure, then we remained on their own because HIV does not have a cure. Yes, society is still what it is, of course. Let's not fool ourselves. Sometimes it's true that when we run this kind of speeches, it seems that we researchers or the industry has different features from society. No, we're all the same. To remove a part of the responsibility of industry, let's say that the EMA has plays a very important role in constraints. Sometimes the regulator, the EMA more than the FDA, by the way, puts certain constraints, because we've talked about improving dosing for pediatric patients much more quickly to go directly to phase 3 straight away, and this would shorten very much the length of these studies. And research groups, in the case of HIV, which is what I have devoted most of my career to, this community advisory boards have been involved from the pre-design phases, and that's something that we are very clear about. Not all researchers, but we do, at least. It is indispensable, for instance, in curing trials, and very often when you want to publish a paper, you're asked if a community advisory board was involved. Of course, you need to have the approval by the ethics committee or by the regulator, but we could also add the mandatoriness of having these community advisory boards. In the UK, you always know that in the protocols there is a member of the community as a member of the protocol design team, and that person helps review materials, because what happens to Joan, if you don't have, for instance, an informed consent, very often the ethics committee would pass it, but the language is, well, who read this? You wonder who read this, because it's impossible to tell this to a person with HIV. It just doesn't make any sense, and you need to do that from the very beginning. And there are two ways. Either you create these advisory boards and you discuss the protocols before you send them to science, and then they already have this additional clarity. And if you talk to the promoter and you tell them that they should do that from the very onset, very often they do. And if not, they do it afterwards. And that means that things will get much longer. Of course, but it needs to be people who are very well trained. And in the part of children and teenagers, and especially in the case of parents, it is much more complex. And it's important to bear that in mind. It's simpler in chronic diseases, as you mentioned, but we need to have the involvement of these people. We need to train these groups of experts, because there are certain diseases where there's already a long-standing tradition on that, but in pediatrics, that's not very often the case. So one of our roles is to train not expert patients, but citizens who are experts in healthcare. My name is Montse Barceló from IRISCAT. And I think we are currently at a very unique moment. The industry is thinking about patient centricity, and patient is all over the place. And that was not the case in the past. I think that many patient associations, and even yourselves, the clinical centers, because you see those protocols coming in and then things are being delayed. Very often we are not concluding our trials on time because, there aren't as many patients as we thought there would be, or they just don't want to join. And I think that AI is also very helpful, because with everything that is published, we manage to identify patients, associations, and centers that were that could not be identified in the past. Sometimes it is very hard to reach the 19 patients you need who are scattered around the world for a rare disease. But nowadays you have platforms and companies who tell us what they can do for us, and you can really reach out to many other people. So it's a unique moment. It's very timely, and I think industry listens to KOLs and researchers much more than patient associations. And of course, I think that you play a very relevant role. We can't force them. It's not in the regulation, but you can say that this is critical for you and that the protocol needs to be reviewed by patients. We were in the World Orphan Congress, and those citizens and patients, some of them have even created companies. Many of them are American, of course, because their children are suffering a certain disease and they know, well, as much as any KOL or any of those who are in clinical trials. So I think that this is a very unique opportunity we have where patient centricity can become a reality. We don't want to be centered, the patients say. We don't want to be used. We want to sit at the table. That's what they say, on an equal footing with the rest. And that's what we really want to achieve. And I think that this is a very unique time.
Thank you very much, Montse. I do agree. And I also have a question, because sometimes it's difficult to find people who want to have a voice, because very often, and I have worked in different projects where there is a clear voice, but in other diseases, sometimes you don't have that voice. Well, I think that this is just what Pere was saying. It's true that chronic diseases have had a longer tradition of having associations. So maybe we have to go beyond the patients in the sense that there should be citizen associations of people who have this background in research and who know the procedures and who know what a procedure means, what a consent form means, whether everything is correct or not, whether if the procedure has been thought for a diabetes patient, and not just from the perspective of the promoter, because sometimes patients' associations are really useful, but we don't always have patients' associations for every disease. Pediatrics is a clear example, yes, and we have to bear in mind, and we work very much with associations, and the effort that we ask from parents of seriously ill patients or chronically ill patients, and these parents have to work, of course, and we're asking an extra job from them. Of course, if you meet with them at 8 o'clock in the evening, in our case, this is overtime because it's our job, but for them, it's an extra job, and it has to be a quality job, as Joan just said, and sometimes this is complicated. I remember working with the mother of a foundation. For her, this is an additional job. She chairs this association, and this is an additional job for her. And it's difficult to have this relationship, and it's sometimes that is also difficult to maintain through time, even in chronic diseases. Of course, training is key, but we need something that's more general. I'd also like to mention the elephant in the room based on what Petra has just said. We think that this should be a remunerated job. It should be remunerated. And every time a pharma company came to say, could you review this protocol for me? We would say it's 200 euros. It's much less money than you would pay the principal investigator. The money stays in the foundation of the association, but why should I do this for free if this is a medication that will be marketed? If it's an observational trial, that's different. But if it's a private promoter, pay me. Of course, I'm providing you this added value and quality. Just add me in your budget. It's just going to be a drop in the ocean. And this motivates people because their job is valued professionally. And with regard to what you have just said, do you think that things would change then from a regulatory standpoint? Do you think that things would change if institutions would force to have this perspective in the design of trials? Well, the ethics committees do force you when you have some posters or any materials to recruit patients for a clinical trial. All of that needs to be reviewed by an ethics committee to make sure that the message is appropriate and that there are no false expectations. This is well regulated, but I don't know whether these ethics committee are guaranteeing the community perspective, because if this is not stated by the regulations, then we just don't do it. If you don't have to check that box, then very often you just don't do it. So very often you cannot demand this if it's not feasible, because if there's no ethics committee, then of course it's impossible. However, it would be good to create these committees because they will be demanded, because very often the biosafety committees in hospitals were created in this way because it was required by the regulations. So overnight you had a new biosafety committee or board at the hospital. So maybe this would be the way to do things, but for some therapeutic areas, they exist, but this is not really requested. I think that the main thing is to provide a quality response by having a health care type of training, and it should be a remunerated type of activity. The same happens with, for example, with you create a patient association. This gives you extra points when you want to access to some financing. Look at our center or many other hospitals that do take this into account, but there's a lot of work to be done. But it's true that the public administration has started to be involved, and they're even leading some of these work packages to obtain financing for these associations. And this is less regulated, however, in the private research. First of all, however, we need to have a society that is aware of the different aspects of healthcare. We are far from that. Well, as you mentioned, there are also networks and AI, and that seems very valuable to achieve these goals. But then I don't really know whether you have seen a change in the new generations, whether they want to be more involved in research. I don't know. Because in my field, I haven't seen any changes. 20 years ago, patients were never taken into account. Well, in pediatrics, it's obvious that there has been a change in this regard, but it varies greatly depending on the type of the population involved. If they are minority diseases that are associated with certain ethnicities that have a lower socioeconomic status, then it's more difficult to involve them. And even if you have an association, there is a very low representation of this population, but at least it's a step forward. But there are other diseases with other characteristics where the population has a greater socioeconomic capacity, and then you have a better response. In any case,
Yo creo que estamos ahí.
The pace we have today is not the best pace, and we would need to change things. As Bea was saying, it should be required by the regulations, by the institution. Well, I think
En ese sentido, has puesto un poquito
in the case of HIV,
por ejemplo, la comunidad del
in the
VIH.
case of the HIV community, we do discuss this
No, yo soy un dinosaurio.
generation, this new generation replacement. I am a dinosaur. And we also have active policies to involve new members with mentoring processes, with support and accompaniment processes. But it's not easy to include the new generations, to involve them, because when you have to respond to a protocol, those who have been around for 15 years know more. But of course, the new ones have these new, fresh eyes, and they can see things from a different perspective, because I've been around for so long that I always look at the same things. I pay attention to the same four things, whereas new with your fresh eyes can see new things. So we should give value to the new generation. The fact that they're younger doesn't mean that their perspective is not valid. In fact, they can provide us with a perspective that we cannot offer, and they're also more connected to certain things. For example, I don't go out at night anymore, so they do. They go out at night.
Ellos sí.
There are many topics regarding clinical trials where we focus on the importance of real life habits. Real life habits. And I do believe that the new generations have helped us understand that our narrative cannot be a good, bad patient narrative. Every patient should accept their own reality, and we should support them as much as possible. And this is something that the new generation understands very well. We have referred to the regulatory aspects and the new generations and their perspective. I don't know whether there are any other questions from the floor. Don't be shy. Well, my opinion is not the opinion of the company I work for, but I do agree that sometimes it's difficult to change things when things have always been done in the same way. And it is true that now we're starting to include the patient, but not as much as we should. And we are not giving them all the importance we should be giving them. We're just following the trend. But it is true that the regulatory part by the agencies is important, and it is the agencies that asked us in the past to introduce many changes, and I do believe that including patients from the very beginning will save time in many cases, and we should also be broadening the population group, because it's often true that the drugs are addressed to a specific group population, but you are not allowed to treat that population group until you reach a certain point of the research. And in the case of vaccines, it's very clear. We are treating healthy patients, and those vaccines, however, are for those who are in a situation that with underlying conditions. So we need to see how these treatments work on healthy individuals. But of course, if these drugs are addressed to a specific population with a specific disease, we should be able to access them because we are referring to patients with very specific characteristics. So we need some guidance also in this regard. And the whole process should be made easier. But it is true that there is a lot to be done. One clear example was the HH3 clinical trial. And we were waiting for the vaccine for adults, and this is a clinical trial for children. And the recruitment capacity has been low. We have a delay of about 6 months, and we haven't been able to reach the number of 300 patients for this HH3 clinical trial. Of course, when you need to do a clinical trial in children, you have to take into account all these things. This is a very interesting discussion, and with regard to what you were saying before, Pere, when we're referring to very specific populations, very often we are testing a vaccine in a population that doesn't have these comorbidities or these underlying diseases. Here we are referring to a population with immunodeficiencies, and the drug may not work in this population because the clinical trial has not taken into account the limitations of this group. So this is a vicious cycle. We need data, but the data come from the healthy population, but you need afterwards to adapt that drug to the population that has that disease. If not, the outcome will be suboptimal. However, if we look at the design of the system, you can even reach the wrong conclusion that the vaccine is not working. Maybe it's not that. It's just that you need to adapt it properly to the target population. And this is mainly in vaccines. And of course, this is something we should always bear in mind. And my second remark is, what can we do? What should we do when we design these clinical trials, when we want to include primary immunodeficiency diseases, because the same disease can vary from patient to patient depending on the mutation? How can we control this type of variability? How can we design that clinical trial to obtain results that are robust enough to obtain the best conclusions?
If you allow me, I'll answer that in a second. Regarding the first question, and also partly regarding the second question, one of the most difficult discussions is the risk-benefit ratio in clinical trials. And when we get involved, that decision used to be made without us, without the community. It was the regulator or the company or the ethics committee who would make that decision. But they're talking about our bodies. They're talking about our lives. So surely we have something to say about that, right? And especially in the first HIV times, activists really pushed the idea of taking on more risk because the alternative was dying. And when we had better and better antiretrovirals that were safer and more effective, we started asking the FDA and the EMA to backtrack because the risk level could not be as high as that. And I remember I've been in a meeting with the Spanish medicines agent saying that, and I was lobbying so that a Pfizer protocol would not be applied in Spain because we understood that the risk for naive patients was too high because Pfizer wanted to start in HIV. They wanted to have a phase 2b3 study, and we said that we did not agree with that. So just as we were willing to take on risks at some point, in a different point, we said, hey, the risk-benefit ratio needs to change. And it is very important because the legitimacy of that discussion needs to have at least patients' representatives, at least one more voice. It's a change of clinical paradigm because patients don't have anything to do now and in 1996. And regarding primary immunodeficiencies, we're far from having this high level of being able to select how we treat each patient, even if they have different mutations that act differently. Only the fact that we were understanding immune defects and that we were pulling up patients as those that have a P flaw or an antibody production flaw, blah, 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 we would be one big step ahead. And of course, that would entail including all these patients who are not already who are not yet in these trials. And then pharmacogenomics trials is something that, using pharmacogenomics in trial design is something that is still not there. And let me add that what you're saying is very relevant. In RBDCOV, we decided to do a trial in immunosuppressive status, but there are, I don't know how many hundreds of immunosuppression conditions, and we chose 5. We couldn't choose them all. And how did we select those 5? Well, based on the COVID epidemics, those that were most affected by that, and pooling this different kinds of immunosuppression, for instance, antibody production or another cell immunodeficient that was affected by anti-rejection therapies in transplanted patients, or HIV populations who were excluded in the COVID vaccine trials. So that was a first step. And then when interpreting results, each one of the diseases has a totally different response profile. And if we had not done this trial, that vaccine, we would have said, okay, we would have known that it generated antibodies and that it could be given to people with immunosuppression, and that's all. But the fact we generated those bodies of data was the first step, and it has to be done. It makes design studies more complex. Of course it does. The easiest thing is to go to healthy individuals, but we need to make that effort because we are all different and because those therapies are going to be given to a very heterogeneous population. So the more we know about how that therapy acts on that group of population, the better. And it would be ideal to have specific patterns for each population that was based on how we know we can optimize the effectiveness. Because if we know that for this population with such and such a defect, we can combine this vaccine with this adjuvant, and for a different group, it's better to give 3 doses instead of 2, well, that would be fantastic, but we can only do that if we generate the data. And for that, we need to run the trials in these groups of population. We can't invent, which is what we've been doing so far. And what's most important means it doesn't mean starting from scratch. Once you start determining, for instance, in vaccines, when you start understanding which groups of patients behave separately, differently, or the same way, you can pull them together. So if we know that a HIV patient with more than such and such would behave as the general population, well, let's put them with the general population. Or a 12-year-old who has a normal pubertal development, then behaves as an adult, well, then put the threshold at 12 instead of at 18 years of age, and so on and so forth. If you can include that person in the population of healthy patients because they behave in the same way, well, go ahead. Because otherwise you would have the feeling that you're multiplying the duration of the study 20 times, but it's not the case. If you know that the renal transplant patient will behave differently and the HIV is going to behave the same as the general population, well, that's enough. Put him there in the same trial for vaccines. Or we know that in non-defining AIDS traits, even in HIV-positive patients, they don't behave the same as in the general population. Well, okay, then you need to require that from the regulator. It will be more expensive, but it's very important because that population precisely needs many thousands of patients who need to know if that drug is going to work or if the dose needs to be higher and what would be the tolerance threshold and so on, because they, in our community, cohorts are aging, and we know that cancers are more prevalent in HIV patients or AIDS patients, so we need more research. And something funny, we're talking about vaccines and people with immunosuppression. We always think about efficacy. They're not going to be as efficacious, as effective. And with Luis, we were surprised with the RBDCOV. Not even tolerance is the same in different groups that we have included in the trial. We don't see the same side effects profile in the groups. So when you have the prospect and you say, well, the vaccine will do this, and that, but where did you test that vaccine? Because if you give it to a group of population, then the local pain that would be such and such would behave differently in such a group. So if you don't have the data, if you don't look into it, you will never know. And as prescribers of drugs, we do need to be able to say and to explain what will be the side effects of the drug in that population. Not only efficacy, which is what we want, but of course, safety and tolerability need to be well studied. So it's so obvious that we need to do that. And even if it's more costly or more cumbersome, we need to do it anyway. I think it's a really interesting discussion. I would continue with this very keenly, but I think we're coming to the end of this talk, and I think we've really done a good job in defining how we can make clinical trials more inclusive, and we will have to think this much more thoroughly. I would like to thank the three of you, Joan, Beatriz, Pere, and also Petra, of course, for this conversation. It's been a very enriching conversation. I hope it has been for you too. And that's about it. Thank you very much for having come to the RBDCOV Science Café, and I hope we'll be able to meet in other similar events. Thank you so much.
En otros eventos parecidos.
Next time with a coffee, though.
La próxima con café.
Thank you very much.
Thank you very much for being here. Thank you.
Molt rebé. Perquè de 5 anys quedaran un off the record.
Exacte.
Bueno, un plaer parlar amb vosaltres.